where do I go? Apparently we're ready. Oh my God, Doryen, are you ready? All right, where do I go here? My mic is on? Oh. Apparently my mic is on. Shall we get started? All right, thank you everyone for coming here. Welcome everyone, in person and also online. There's a few things that I just have to quickly dispense with. If there's a fire alarm, go out of here, especially you people watching online. The assembly point is at Dangle Plaza. The event's being recorded, and if you are watching it online, you can still ask us questions, and Bernadette at the front here will relay those questions over to us. Professor Doryen Bubeck grew up in upstate New York, and her academic journey began when she was studying at Rensselaer Polytechnic Institute, where she did an undergraduate project with Joachim Frank, using electron microscopes to visualize the actual molecular shapes of things that she had previously only seen in textbooks as little cartoons. And that was the formative experience that she said shaped the rest of her career. Also, of course, some of you here in this audience will be feeling right now that we have undergraduate students in our labs doing their projects. And that does feel overwhelming, and yet it actually has the power to change people's lives, as it did, I think, for Doryen. Joachim went on to share the Nobel Prize in Chemistry in 2017 with Jacques Debauchet and Richard Henderson for their work in using electron microscopes to visualize molecules, and we're delighted that Richard is here tonight with us, and he'll give the vote of thanks at the end. Doryen followed this up with a prestigious fellowship from the National Science Foundation, pursued her PhD in biophysics at Harvard University. She then felt finally that she needed to get out of the east coast of the USA and decided, thank goodness, to come to the UK to work at the University of Oxford, working with Yvonne Jones, where she also held prestigious fellowships from EMBO and the Cancer Research Institute. At that time, also, she felt that Suzanne Lee at Oxford University was an inspiration to her. And in Doryen's words, it was because Susan Lee enabled others to be great. In 2012, Doryen joined us here at Imperial College London as a lecturer in our Department of Life Sciences. No coincidence whatsoever, but that's the same time I left Imperial College London. Doryen won a Career Establishment Award from Cancer Research UK, and her rise since then has been fantastic, culminating in what we're celebrating today, her achievement of becoming a Professor of Structural Immunology in 2023. Her lab studies essentially how the immune system works and how it fights pathogens on an atomic scale. In an interview with her that I've recently just watched, she said she loves the small discoveries that happen every day, because it's those tiny puzzles that then accumulate and then open up the answers to bigger questions. beyond her own research, she's also played a big role in shaping all of the community that we have in science. She was the director of the Center for Structural Biology, helping that center thrive, keeping it having state-of-the-art equipment, championing everything that it does, including all kinds of workshops that are equally formative in people's lives. And right now, she is the director of research for our Department of Life Sciences. And so I've got to personally work with her much more closely because of that role. And we have meetings where there are complicated decisions to be made, and Doryen is absolutely brilliant at thinking very clearly through a problem and stating what we should do about it. Every time there's a difficult decision for us to make in our department, Doryen is someone that you can rely on to listen to and have good ideas about what we should do about it. Also, what's quite inspiring about how Doryen works is I think she's always open to exploring new and different and interesting things. The most recent is that she's part of a sort of large consortium of scientists from across the whole country that have together been awarded £6 million to look at how pathogens cause crop diseases. Finally, in a recent interview with some of our students for a podcast, Doryen was asked, what advice does she have for young scientists? She replied, follow your passion, be flexible, and try to find people who will open doors for you. I think Doryen is going to open doors for us tonight. Please join me in welcoming Professor Doryen Bubeck.
Great. Thank you so much, Dan, for that really kind introduction. You know, a lot of people think that science is all about big eureka moments that are captured by fancy journal articles. And I'm just going to let you in on a little secret. That's not true. Science is all about a collection of stories about people who have influenced us and who we have influenced to think about the world in a new way. And tonight I'm going to tell you my story. I'm going to tell you about some of the big questions that have inspired me to understand how tiny machines work inside cells. So what are proteins? What are tiny machines? So proteins are the world's tiniest machines. So they are, if you think about the size of a pound coin, they are 0.000001 times the size of a pound coin. If DNA is the information content of your cell, the proteins are what do all the work. And there are millions of different protein machines inside a cell, and each one has a special job to do. They come in all different shapes and sizes. And really, by trying to understand what their shape is, we can try to understand what their job is inside the cell. And if you kind of think about other machinery that maybe you're more familiar with, like construction machinery or things like that, you see a giant digger with a trough, and it makes you ask the question, well, what's it pushing? And it's the same thing for structural biology. So we use electron microscopes, and what they do is they let us see proteins in atomic detail, or really, really high resolution. Now, a lot of you may not be familiar with electron microscopes, but they're really similar to light microscopes. So both electron and light microscopes, they have a source that is going to be emitting waves. They both have lenses that need to focus these waves, and these waves interact with the sample to make them scatter these waves, and the images are recorded on detectors or cameras. But one really important difference between electron microscopes and light microscopes is the wavelength of the electron beam is so small that it allows us to look at very high resolution or a lot finer detail these protein structures. And so although they're really powerful machines in that they allow us to see these molecules in very high resolution, the images that we collect on the microscope are really noisy. And that's because one, not all of the electrons are going to interact with the sample. Some of them are just going to pass straight through to the detector. And then the ones that are interacting with the sample are kind of grouped into two categories. You've got elastically scattered electrons that once they encounter the sample are going to be scattered, but energy is conserved, and those ones count towards the signal. And you have inelastically scattered electrons that interact with the sample, lose energy, and they're essentially contributing to your noise. So to illustrate this point, I need some volunteers from the audience. So Dan has already agreed. He's been a super good sport, Dan. So come on up. And so Dan is going to be the sample in my demonstration of what happens in an electron microscope. So you need some PPE, Dan.
Okay, great. I don't remember agreeing to this, do I?
It was at that lunch, remember?
Oh, yeah, okay.
And I'm going to need some more help from the audience. So now I'd like to invite Chiara, who is an amazing artist who I've commissioned to make an illustration shield of a protein complex that you're going to be seeing later in the talk. But she is well, she's not going to represent. She's going to hand over the shield to Dan. Thank you, Chiara. Let's give Chiara a hand. And Dan, you're going to need these as well.
What is that?
Okay, they're ear defenders. Okay, now I'm going to need some more help from the audience. I need 2 volunteers for my magical green cloth. I need some volunteers from the audience, please. Yep, okay. One there, one more. This one here, yeah. Yeah, you. Okay, so Dan, you got to stand here.
Okay. I can't hear you.
That's fine. Okay, so you 2 ladies, please don't fall down. I did a risk assessment for this demo, so stand one on each chair. No, you got to stand on the chair. And don't fall. Okay, and hold up this green cloth, and you're going to want to make sure that you're not exposed. And then, okay, so now I need 5 more helpers. So I've got my 2 helpers, my bestest helpers, Aldi and Araya. Come on up. And I need 3 more helpers from the audience. Yep, I got the one in the back. Yep. And 2 more helpers. Yep, one in the back. And no, I'm going to go to the little boy here in the front. Okay, so come over here, my helpers. Now, you're going to hit the target, yeah? Hit the target or miss the target? Hit. Aldi, are you going to miss? Okay. You are going to miss the target. Don't hit Dan, okay? Don't hit Dan. Do hit Dan. Do hit Dan. Do hit Dan, okay? Okay. Now, I've got my amazing helpers here who are going to be bombarding Dan with a beam of electrons. And in my beam of electrons, I've got elastically scattered electrons in gray, and every other color are inelastically scattered electrons. Noise, signal. Now, I need Georgina. You're going to sit in your chair because you're in the perfect spot. So you are going to count for me every ball that does not hit Dan. Okay.
Wait, what? How many balls are going to hit Dan?
And now I need 2 more volunteers in the audience. I'm going to go Costas. You are going to count all the gray balls that hit Dan. And I need 1 more volunteer. I'm going to go Rivka. You're going to count all the colored balls that hit Dan, okay? And you're going to remember those numbers because I'm going to ask you at the end, okay? So be alert. You've got 1 minute and 10 seconds of bombardment. Okay.
That's really great. I'm thrilled.
All right, you guys know what you're doing. You know what you're doing. Okay, ready? Go close there and do until the balls are empty.
Wait, so I'm
You are blocking.
blocking.
Yes, you're blocking. Ready?
Okay,
3.
wait.
Are you ready, Dan?
I'm ready.
Okay, 2, 1, go.
Oh my days, what is this? This is awful. Oh, agony. Can I throw some back? Ow! This is awful. What is this? Am I allowed to hit them?
No, keep going. You got to finish my basket. Keep going, keep going.
Ah! It's like broken my teeth. What is this? Oh, I'm bleeding.
Okay, are you guys done? Oh my God.
Oh,
All right.
God.
Give them a round of applause. Thank you so much, everyone. You guys are awesome. Go sit down. Thank you so much, guys. Thank you. Thank you.
Well, I've got a swollen lip. I'm bleeding. I've got a headache.
Thank you very much, Dana. Go sit
The
down.
painting's good.
Thank you very much. Please don't trip when you get down. Okay. Now, who is counting George? How many did not hit Dan? 64. Oh, these throwers were not as... So 64 did not hit Dan. How many gray ones hit Dan? 22. And Rivka?
I got 56.
Yeah, that's... Yeah, the missers maybe needed to miss more, but... But you kind of get the point. You get the point. So most of the electrons aren't actually going to hit Dan. They're going to go straight through. And because that signal is so big, it swamps out the signal that actually interacts with the sample. And then out of the electrons that are hitting Dan, only a third of those really are the ones that are contributing to the signal. The other ones are contributing to the noise. And then degrading the quality of the image. So I hope you now fully appreciate how an electron microscope works. Okay, now I've totally lost my clicker. Okay. Okay. Okay, so as Dan mentioned, I first got introduced to what an electron microscope was actually as a high school student. So I joined Joachim Frank's lab when I was 16 years old and was interested, well, at the time I didn't know what I was interested in, but he was doing amazing work looking at how ribosomes make proteins that are destined for the membrane in this case, so through the secretory pathway. And one of the things that was so inspiring to me at the time was the fact that you could see these proteins in 3D. You could put on these big 3D glasses and look at that silicon graphics computer and see those molecules rotating in 3D. And this is just a movie made in Joachim's lab around the same time that I worked there to give you an idea of what some of these images look like. And just to put it in perspective, this is coming from a time when, I mean, I don't know how many people in the audience had a Commodore 64, but like that was my computer at the time. And this was just a game changer. And then I got the bug and decided that I wanted to understand what the structures of proteins were. And I was particularly interested in how the immune system works, this battle between host and pathogens. And so I wanted to explore how viruses got inside cells. And I went to Boston to the Harvard Biophysics program. And there I met two amazing scientists, Jim Hogel and Dave Filman. And these guys were really inspirational because they not only taught me how to solve science puzzles, but they also taught me how to lead a team. One of the best things about Jim is that he really thinks about what does that person want to achieve, and tries to do everything he can to try to help them to do that, no matter what it is. And for me, I wanted to do cryo-EM. And no one was doing cryo-EM in his lab. It was a pretty kind of nascent, like niche technique. And so what Jim did was he helped me to do cryo-EM to try to understand how poliovirus attaches on to cells. And this was a really hard problem at the time. And Jim wanted me to say tonight that like in his grant application that reviewers said that this project couldn't be done, and if they could be done, it couldn't be done by me. And with the support of Jim and Dave Philman, and actually the cryo-EM community at a Gordon conference was able to tackle this problem. And the structure here that is rotating around was actually even made into a sculpture and was on display at the Smithsonian Institute in Washington, D.C. for a commemoration of the poliovirus vaccine. So after I finished my PhD, I wanted to do a postdoc. I wanted to think about this battle between host and pathogens, but from the other side, on the human immune system. And so I went to Oxford to work with Yvonne Jones, who amazingly is in the audience here this evening. And in Yvonne's lab, she really taught me how to broaden my toolkit for structural biology. I worked on lots and lots of different projects, and really all centered around trying to understand how the immune system recognizes these challenges. And that's when I met Susan Lee, who introduced me to the complement system, and trying to understand how the immune system fights bacteria. So what is the complement system? So again, a lot of you may not have heard of complement, and in terms of thinking about immunology, you might think of cell-based immune systems like T cells or B cells or natural killer cells. But complement's a little bit different in that it's a protein-based immune system. So it's a protein-based immune network. So remember all those tiny machines, they're floating around inside your body all the time, and they're constantly on surveillance for when it's going to be detecting a pathogen. And this kind of very complex and intricate network of proteins, it really requires the correct coordination of all these different players. And if there's anything that goes wrong in those pathways, it can lead to lots of devastating consequences for human disease, and with a wide range, ranging from blood clots to recurrent infections, immune evasion mechanisms by cancer, bursting of red blood cells, and also transplant rejection. So it's really important to understand not only how complement fights infection, but also how your cells are protected from damage. And that'll come back again later in the talk. So the main area of the research interest of my lab and is really focused on trying to understand how the membrane attack complex works. So the membrane attack complex kind of does what it says on the tin. It attacks membranes and punches holes in target cells. And it's the direct killing arm of complement. And it's a very, very effective killing machine against gram-negative bacteria and also viruses. So the first big question that I was interested in answering is, well, how do these MAC pores form? So this is an electron microscopy image taken, I think, about 4 years after I was born. So the first images of the membrane attack complex kind of came to light around 1982. But if you notice, this isn't particularly high resolution. There's not a lot of detail there. So it kind of it had a big gap in the field. And until kind of the early 2000s, scientists didn't really know how to tackle it. And in the early 2000s, structural biology, specifically using X-ray crystallography, which is another kind of structural technique, they kind of broke it up into little pieces that were a little bit more tractable. So they tried to solve they did solve the high-resolution structures of lots of different bits of this membrane attack complex. So you kind of had pieces of this puzzle, but you didn't really know how all the pieces fit together to make this big pore. And this is where I came in. So I joined Imperial in 2012. I had lots of big ideas, lots of big questions I wanted to solve, lots of ideas, but no money. So I needed to write my grants, and I remember, I just want to tell this story. I went for my Cancer Research UK Career Establishment Award interview, 9 months pregnant. And I went in for that award, and I totally did not think I got it. And I remember going home super, super upset, telling my husband, oh, it's horrible, I didn't get this award. And he knew this was like mission critical, that even the emergency cake hotline was not going to fix this situation. So what did he do? But he went to Hamleys and bought a remote-controlled helicopter to then cheer me up by launching this helicopter off my enormous baby bump. So I just want to say, without his support through this whole thing, I wouldn't be able to tell you all of the exciting stories that I'm going to tell you. But luckily, I did get that money, and I was able to start my team. And here's just a picture of us in the beginning part of the lab, and also Aurea after I came back from maternity leave. And you didn't see this, Paul, on the microscope, where Elmo is helping out the science experiment. Okay, so the next story I want to tell is a little bit about Marina Serna. So Marina Serna was my first postdoc in the lab, and she had the most amazing combination of creativity and meticulousness that you need in a scientist. And it's because of her that we were able to solve the first structure of the membrane attack complex. And I should also say that this was a really exciting time for the field of cryo-EM. There were huge technical innovations in microscopes, in detectors, and there wasn't very many of them around. And part of the reason we were able to solve this structure and have this big impact was thanks to Richard, who gave us a chance and gave us some access to the microscopes at the LMB and also to Diamond Light Source. We were one of the first users at EBIC to solve this structure. And that really opened the door for what we could achieve then for this structure. So now we were able to see how these different proteins came together to form this giant membrane attack complex and how they made up the pore. But I like to show this picture because one thing that's really important here is always looking for what you can't see. So these proteins here were assembled on a membrane and then extracted out of that. And one of the interesting things, I don't know where my clicker is, but where the arrow is here, is where that pore would interact with the membrane. It really made me think about, well, what are these protein-lipid interactions that are driving the formation of the pore? So, does it go to the next slide? Okay. So we published that structure just in time before the arrival of my son, Alden, who, when I was coming back from maternity leave, again, microscope time was still pretty tight and scarce. So he would be my microscope buddy and accompany me to Diamond Light Source and drool all over my ID badge. But we had to I rebuilt the team after coming back from maternity leave. And one of the things I wanted to say is the next slide is really a team effort that bridged Marina's work, Courtney's work, and the work of Anais Meni in the lab, another postdoc. So by collecting lots and lots more data, we were able to take advantage of new advances in computational image processing tools that allowed us to separate out different kind of conformations of these proteins. Because it turns out, I mean, it's not really something so unexpected, but machines are dynamic, right? They need to move to do their job. And so proteins are the same way. And so by using these computational tools, we were able to separate out these different populations of particles that allowed us to get high-resolution images or reconstructions or maps for each of these two states. And why that was important was because that then allowed us to really see kind of the chemical interactions between these different proteins. And I know I'm not going to go into the details on what a lot of these colors mean, but you can kind of judge by the dotted circle in this kind of S-shaped blue bit that charged patch is going to interact with the lipids in a slightly different way. And so one of the things that Anais did was really trying to integrate the structural biology with membrane biophysics to try to understand how these complement proteins prime that membrane for eventually needing to punch the hole. So then COVID hit, and the whole world changed again. And the only science experiments I was really able to do was at home, making my two kids do fun science Fridays in our backyard. But I want to say that our lab was fortunate enough that we were able to pivot a lot of our work to computational approaches to process all that data. We've got Emma and Thomas in the lab, that were in the lab in the audience tonight, and they were able to pivot their projects to take kind of new and be flexible, take new approaches to try to keep going, really. And I just want to say that I'm really proud of the lab for supporting each other through that hard time, right? It was a hard time for everybody. So most of the work coming up to this point was really thinking about how does that MAC pore assemble? How does it punch holes in target cells? But really, if I think about some of the big questions that are motivating me, it's about this host pathogen interaction. How are my cells protected when the immune system switches on? Because there's no, the MAC is a really effective killing machine. There's no specific receptor that activates it. So my cells would be very susceptible if they weren't protected. And so this story here is really a story of David and Goliath, where Goliath is that giant beta barrel immune pore of the MAC. And David here is this small little GPI-anchored cell surface protein on your cells. So how does David battle the wrath of Goliath? That was the next big question that I was interested in answering. So first I want to introduce you to David. So David is CD59. So CD59, again, is this small GPI-anchored, surface-anchored glycoprotein ubiquitously expressed on lots of your cells. And so this is your body's last line of defense against that giant beta barrel pore. So these complement proteins, when they're assembling on the cell surface, are then blocked at 2 different points by CD59. So I was interested in trying to understand how CD59 is doing that. So in order to catch MAC, I had to set the trap, right? So part of all of these studies are about designing the model membrane system that will support the biology that I want to study. So we needed to set the trap for the membrane attack complex. So I want to just share that this is the work by, a lot of work kind of went into developing that model membrane system by postdoc Nita in the lab and my PhD student Courtney that worked on these lipid nanodiscs. So these are these membrane discs that we decorated with kind of a synthetic version of a GPI anchor to stick CD59 on there. And so a lot of work went into kind of developing the experimental system to set that trap. And then we activated complement on these little lipid discs, and we're able to stop the membrane attack complex at 2 different points in its assembly process. So this is largely the work of Emma Kouvs, who's in the audience, and Scott Gardner, who I think is online. And so we used cryo-electron microscopy to solve the structures of these 2 different complexes. And the main kind of key take-home message that I want to share with you about that is that when the membrane attack complex forms the pore, it does it by having these kind of coiled or helical residues that need to undergo a really big conformational change. So they go from like a coiled shape to like a knife, right? So they unfurl to make a beta hairpin, or like a knife that's got to slice through that lipid bilayer. And what CD59 does is it's in the right place at the right time. It's lying in wait in that trap. And it's waiting for that coil to unfurl, and it catches the hairpins as they are cascading towards the membrane. And it kind of deflects them away from that membrane trajectory. So it catches them, and then it deflects them away so that the knife can't slice through that lipid bilayer anymore. So it catches and deflects them. And it kind of does the same thing for the 2 different proteins that it binds to in that membrane attack complex. So then in 2023, I was diagnosed with breast cancer. And this was my chemotherapy sticker chart. And it is an extremely powerful image for 3 reasons. One, it reminds me that no matter how big the challenge is, if you break it up into small little pieces and just keep going, then you can get there. And second thing it reminds me of is that in order to achieve something really hard, you can't do it by yourself. And you need to surround yourself with people that are going to help you get there. And the third thing that it reminds me is just how important the basic science research we do in the lab every day is for opening up the next new cancer therapy, opening up the next new antibiotic. It's about how the work that we do in the lab every day is going to unlock that new challenge that's going to help people. And so with that, I want to pose that challenge to you. What is your next big question? You got to pick the most important, biggest, impactful question that you can think of that's going to help the most people. And don't be scared if it feels too big, because if it wasn't big, it wasn't worth doing. And you will get there if you just remember that you got to break it up into small pieces and just keep going and surround yourself with amazing people that are going to help you get there. And with that, I would like to thank all of the amazing people that have helped me get here. I would like to thank all the people who are online and in the audience tonight who have helped me get to this very spot. From Joachim Frank to introducing me to cryo-EM, to Jim Hogel and Dave Philman for inspiring me to be a good scientist. For Yvonne and for Susan, for all the people in my lab, past and present, and the funding bodies who took a chance. Thank you very much. I'm happy to take questions.
Thank you, Doryen, for a wonderful, wonderful, powerful, brilliant lecture. Any questions? Any questions at all? So, so please wait for the microphone to come to you. We'll start there with Mark.
Sorry, just a technical question. I was curious what that little cyan mini kind of beta barrel in the middle of on the side of the pore sticking in did.
Yeah, so that is a good question, actually. So people don't really know. That protein is called C8 gamma. And in the membrane attack complex, C8 is a heterotrimeric protein. It has two pore-forming proteins, two pore-forming domains, and then that lipokaolin gamma. And in the soluble structure, gamma blocks the binding site for the next component. So when the C8 joins the pore, gamma rotates out of the way and opens up the next binding site. So I think it's like a regulatory mechanism.
Thanks, Doryen. Yes, just next to it, yeah, same row.
Hi, Doryen.
Hi.
Fantastic lecture. I'm curious, where do you go now with that? Now you've worked out C59 and how that works, and the MAC complex, you've solved that. So what direction do you go now with that complex?
Yeah, so you got to go bigger, right? So you got to think about what is, and this is like structural biology all across the globe. It's kind of thinking about structural cell biology, right? So what are the cellular consequences of that CD59 blocking that MAC complex? So does it get internalized? Where does it go? What other signaling pathways are they activating? Because if you think back to that slide that was talking about all the different disease pathologies for complement, it's because complement has crosstalk with so many other cellular pathways. So it's not just about immune recognition on the surface, but those immune recognition events have knock-on consequences below the plasma membrane. And so I think that's the next challenge for that.
Thank you.
Yeah, up there.
Hi, Doryen. That was terrific. It's just, I guess, a kind of functional question. So you showed that image, the little movie from the Many paper, where you had the pore complex, and it was kind of, looked like a ring that was opening and closing,
Yes.
right? So what's the functional significance of that opening and closing?
So I can just guess, but I think the dynamic nature of opening and closing that pore probably has a role in disturbing and kind of adding stresses to the outer membrane of the gram-negative bacteria. I think that, you know, if you have something that's constantly moving like this, it's going to be working its way to disrupt and cause large-scale disruption.
Yeah.
So why have bacteria has not evolved a David of their own, I guess?
Yeah, there are loads of other immune evasion mechanisms that bacteria have evolved to block complement, and we're working on lots of different parts of that in the lab as well. So there are lots of ways that bacteria kind of hijack complement regulators in other ways.
You're just 4 rows down the same. No, thanks. Yeah.
Lovely talk, Doryen. Yeah, thank you very much. I don't know much about these systems, but I was wondering, when you see that ring, it was actually bigger than I expected. So it's like, it's not as tiny a machine as I expected. It's kind of a big machine.
That one is a big machine.
And I guess I was wondering, like, why do you need such a big ring? Like, is it just it just takes that much force, or like, why does it need to be so big?
Yeah, I don't know why it needs to be so big. Yeah, I don't know. I don't know. I don't know.
Yeah, Mike.
Doryen, a model inaugural lecture. We've got Richard here, who pioneered EM. You who are doing it now. Where do you think we'll be in 10 and 15 years with cryo-EM?
I think machine learning is going to have a huge impact in data mining, as Pierluigi in the audience will definitely know. I think where it's going to go is improving sensitivity for detection in cellular images, or improvements of contrast in collecting cellular slices, and then machine learning algorithms that are going to be able to identify properly smaller and smaller objects in those things. It's the kind of contextual information that I think that's where the field is moving.
Any more questions? Bernadette, is there questions online?
No, Dan, no questions, but some lovely comments that you can look at later.
If anyone does have a question online, please feed them in, and Bernadette will wave at me when.
I will do that, but actually maybe I can just come in with one comment.
Yeah.
Somebody's dad, I think, whose son is friends with yours, has left some nice comments, which I thought was lovely. So I checked in. So, but yeah, no questions at the moment. Thanks, Dan.
Yep, please.
Thank you for your lecture. I was wondering,
Where
what is the next?
person? Okay, I see.
What is the next big question for you in your lab?
Well, in the immediate term, because I'm always really interested in this battle between hosts and pathogens, and Dan had alluded to in his introduction that I'm really interested in thinking about how that plays out in plant host microbe systems, and thinking about climate change resilience, and how can I use some of the insights, tools, and kind of just knowing how these systems work in the human immune system, how can I apply that now to a completely different scenario and provide maybe a fresh and different perspective on that question?
Yep, at the back, yeah.
Hi, Doryen.
Hey, Sam.
It's been about 16 years since my immunology lectures, but so for the membrane attack complex, is it floating around the cells as a whole complex all the time, or is it
No, it
activated
floats around as individual pieces that are then activated on the membrane.
So there's like a protein on the membrane it tethers to first, and then it all sort of forms a ring around it.
So the convertase, the C5 convertase, will initiate this terminal pathway cascade, with a proteolytic cleavage event that then triggers an irreversible cascade that then means that you can't stop it until you get to CD59.
Cool, thank you.
Yeah.
I didn't quite understand the inelastic and elastic scattering thing. I wonder whether we can repeat the demonstration.
I think you know how it works, Julia. I think you know how that works.
I don't know who that. Please, can we eject? Yes. Go ahead.
Please, could you explain how you were able to predict the mechanism of action of MAC?
Sorry, say that one more time.
Sorry. How were you able to predict the mechanism of action of MAC?
So I think for me, let's take one good example is back to that slide when we were able to model all of the residues, all the amino acids of the membrane attack complex. And then by looking at the chemical properties of those amino acids, what we did was we saw that they had this kind of curved, not planar, charged patch. And so based on looking at the structure and looking at like the biochemical patterning on the surface of the molecule, it made me think, okay, well, that bit, it's got to interact with lipid head groups because those are really charged, right? That's not going to be going through the membrane because the transmembrane bits are all hydrophobic and white. So that then made me think that, I mean, structures don't necessarily tell you the mechanism, but they give you the hypothesis about now what I'm going to test next. And so that structural information gave us the hypothesis that those residues were maybe priming the membrane, interacting with the head groups, bending it in some way that we could then probe with collaborators, Nick Brooks in chemistry, about how that could then change the physical mechanical properties of the lipid bilayer to make it easier to punch the hole.
Bernadette, yeah, online.
Yeah, thanks, Dan. So Sue Gao asks, do you have any special procedures that you use for preparation of your sample for EM?
Yes, so I didn't really talk much about the sample preparation part, just the kind of demo of Dan being pelted with balls. But in the electron microscope, because it operates under vacuum conditions, we have to preserve or protect the biological sample. Otherwise, it will just evaporate and be destroyed. So in order to do that, that was one of the things that Jacques Dubochet won the Nobel Prize for in that shared prize for the cryo plunge freezing method. So the sample is applied to these very small grids, and then the excess liquid is blotted away, and that grid is then plunge frozen into liquid ethane, cooled to liquid nitrogen temperatures. And then everything else needs to operate under liquid nitrogen temperatures to maintain that cryo condition.
Right at the back, yeah.
How did you go from predicting how MAC works to proving how it works?
Well, that was kind of what we were trying to talk about from maybe I wasn't clear in my last answer, was trying to think about how do we then use these membrane biophysics experiments, these complementary assays that then look at bending properties, bending modulus, and that we are able to measure how that changes in response to each of those stepwise additions of the MAC. Yes. Other ways that structural biologists test their hypotheses through mutagenesis, where they knock out different residues and see how that affects on the function.
Yep.
So thanks for the lecture, it was wonderful. I'm going to change tack. It's not a science question, but it's a research culture question. It was so nice to see the young female scientists really obviously flourishing in your lab. Do you want to share some nuggets about the culture in your lab? We always get asked at interviews, so give us some tips.
What do you want me to say? What the culture is in my lab? Oh, you can ask my lab. All the people that are past and present are even here in the audience. I mean, I think the thing for me, and that was really influential in terms of Jim and the way that he approached leading his team, is that I start every catch-up meeting that I have with students and staff with, how can I help you this week, right? You go in with the mind frame that my job is to help you do your job better. but I don't know how to help you, so you tell me what you need, right? And it's that shift of perspective that I think really underpins the research culture in my lab.
One more online, yeah.
So this is a question from Robert Sharrock, who's a zoologist, and he asks, has any work been done at this level of atomistic detail in other organisms, in other animals?
For complement proteins?
Yeah, I think that's what he means.
Yeah, so I would say no, but one really interesting thing that we discovered when we looked at the structure of the CD59 inhibited complex, one of the kind of weird things in the field that no one quite understood why was that, you normally think like human and mouse or human and rat are very well conserved and good model systems for each other. And there's a lot of sequence identity between those proteins. But in the complement system, the rat CD59 won't block human complement. So the binding site isn't the same. But if you model based on the human sequence onto the rat sequence or the mouse sequence for the complement proteins, you can see a co-evolution of the interface residues. So that really showed us that it was this co-evolution of the receptor and the immune proteins that allowed that to work in the rat system or in the human system, but not have a cross-reactivity. So that's the best that I can answer that one.
Thank you.
Okay, Doryen, I think well, actually, I have a question. I have 5 broken teeth, and I wondered if you were a cheap dentist. It was a fantastic lecture. I think that for me personally, I know I've seen 1,000 lectures in my career, like most of us, and that the moment, the sticker sheet, and the fact that we need to pick big challenges, break it down into small pieces, and get people to help us get through it is going to stick with me for a very, very long time. Thank you. I'm going to hand over to Richard Henderson, who now is going to give the vote of thanks. Thank you.
Yeah. All right. Yeah, so it's my job to thank Doryen for giving a wonderful lecture and thank all the audience for coming from Imperial and elsewhere. But I thought I'd just say a couple of things by a kind of reflection on Doryen's style that might be helpful to people who are thinking of going on in research. One is Doryen was very careful always to pick good groups to work with, good colleagues. So first with Joachim Frank and then with Jim Hogel. And then there is one story. When she was thinking of being a postdoc, she went around the world looking at lots of potential places to come, including coming to Cambridge and talking to us. And we did say, Doryen, we would love you to come here, and we offered her, but we were rejected. And instead, she went with Yvonne Jones in Oxford to work on immunology, and then eventually started working on this membrane attack complex. And so we had a second period of contact with Doryen when we were starting to do the cryo-EM with the new technology about 10 years ago now, 2013. Not many people had these instruments. We had one in Cambridge, and we opened it up to a lot of other people. Many people came with really bad samples. Doryen came with a sample, and we looked at the images. We couldn't see anything. We said, are you sure, Doryen, that you...
Richard, your exact words were, why don't you show me where you think your particles are?
Anyway, it turned out she was absolutely right. She could see things that we couldn't see. So she picked a very good problem, and she knew how to get around it. So I think the two things about Doryen that I observed is she's been very good and very careful about choosing where to go and what colleagues to work with. And then she's picked important problems and tackled them when other people thought they were impossible. So I think with those and with this lecture, we realize that Imperial has done a good job in recruiting Doryen here. And I'm sure it's true that even if they don't, Imperial ought to be pleased. They're lucky to have Doryen here. So thank you very much, Doryen. Thank you.
Okay, thank you everyone for coming. Thanks, Doryen, it was phenomenal. Thank you, Richard. We are going to a reception which is in the foyer. You could go out this door or that door, and it's downstairs. Thank you. That's great. Thank you.